Hey guys, welcome to Unit 4. I'm going to be doing 3 lecture videos for this unit. The first video will cover the first 3 topics, and then I'm going to do 2 more videos covering the last 3 topics. So this unit is titled Cell Communication and Cell Cycle, and I'm going to start off with the first topic. But before I do, let me show you guys a list of the topics that are going to be covered in this unit. So there are 6 topics in this unit. For this lecture video, I'm going to be covering 3 topics. I'm going to do cell communication, introduction to cell signal transduction, and signal transduction pathways for this video. All right, so let's start. Cell communication is the process by which cells within a multicellular organism send signals to each other in order to pretty much carry out a unified function. So cell-to-cell communication is a critical function for the survival of cells. And this is responsible for growth and development of multicellular organisms. And there are different ways in which cells communicate. So how do cells actually communicate with each other? There are multiple ways, multiple reasons, different types of signals that are sent, but each type, regardless of the type, it is placed in 3 categories. So it's either going to be a direct contact signal sent, or it's going to be a local signaling, or it's going to be a long distance signaling. So these are the 3 ways in which cells will communicate. And this is very important knowledge for you guys to know. All right, so let's start off with our first way in which cells communicate. We have direct contact, and as the term suggests, a direct contact means that these cells are in direct contact with each other, so they are touching, they're close together. Direct contact is when cells will communicate through pores or spaces between their cell membranes, or in the case of a plant cell, these spaces or gaps are between the cell wall and all the way into the cytoplasm. So in direct contact, the key thing to note is that these cells that are communicating with each other they have to be adjacent to each other, or in very close proximity to the point where they're directly touching each other. The signaling substance or chemical or ligand is dissolved through the plasma membrane, and it passes freely between adjacent cells through these pores. If these are plant cells, the pores in which these messages are passed through are called gap junctions. Please ensure that you remember that. And here's one that you already know. If these are plant cells, then those pores are called plasmodesmata. And in my pictures below right here, I have an example of gap junctions in animal cells. And on this side, I have an example of plasmodesmata in plant cells. All right, so examples of direct contact. So direct contact is very, very essential to immune cells and our immune functions. And or T cells and helper T cells are always going to be roaming or roaming through our bodies, and they're pretty much trying to ensure that all the cells that are in our bodies are good cells, they're not defective, and also they're not foreign agencies, so they actually do belong to your body. And how can they identify that cells belong to your body? Cells that present antigens, which are proteins that are on the surface of these cells. And T cells can pretty much detect these antigens. Now, the only way T cells can actually detect antigens or actually read the antigens is by directly touching these cells with their antigens. So here's an example of an immune T cell that has approached an antigen presenting cell, and that's what cells that have antigens are called. They're called antigen presenting cells. And the helper T cell has approached this antigen presenting cell just to get a closer look to read, to ensure that this antigen or this protein that's on the surface is something that actually belongs to the body, and it's not like a virus or a bacteria or some foreign agent, or the cell itself is not defective. And once it reads it by coming in direct contact with the antigen presenting cell, it can determine whether or not to trigger an immune response or how it's going to be responding. And there are different ways in which the helper T cell could possibly respond. All right, so let's talk about local signals. Local signals are sent from one cell to all the cells within a specific region or a specific tissue. So one cell is responsible for producing the signal or the chemical, and then that signal or chemical will pretty much be broadcast to all the cells that are in close proximity in that region, and we call that local signaling. Now, why is this done? This is done usually, again, with cells that are in close proximity. These cells are usually in the same tissue, and it's pretty much to communicate a message in which the cells have to carry out a unified function. So it could be, for example, and this is a very common example that I will use time and time again, mitotic cell division. So cells within a tissue know that, hey, we need to now multiply and make more of ourselves, or we need to repair ourselves. So like for example, when you go to the gym or when you guys do athletic stuff and your muscles are torn or worn out, the muscles within that specific area can now regenerate, well, not regenerate, but make more of themselves or repair themselves once that signal is sent. So the chemical message will cause a response. So that chemical message will cause a response, and the chemical message is sent to target cells. Examples of these type of signaling are paracrine signaling and also synaptic signaling. And paracrine and synaptic signaling are examples of local regulation or local regulator signals in which these are sent over short distances. So all the cells that are receiving the message are not necessarily in direct contact, they're not touching, but they're in close proximity to each other. And the cells receiving the signals, those cells are called target cells. And here's an example depicted by this picture right here. We have our target cell, and then we have our local regulator cell. This cell produces the message and then sends that message, and that message travels over a short distance, so these two cells are not touching, and then that message will be received by receptor membranes along, or receptor molecules along the membrane of the cell. Paracrine signal is when the signal that is sent, these are usually chemicals. So, for example, growth hormones are sent within the tissues of, within the cells of a tissue. So, paracrine is when regulator chemicals are released. Another example of local signal would be synaptic signaling, and this occurs within neurons um, of the nerve cells. So synaptic signaling is specific to the nerve cells, and neurons are very long cells. So neurons, they pretty much they transmit electrical impulses from one end of the cell, which is the dendrite, to the opposite end of the cell, which is the axon, and then that signal has to be transmitted over pretty much long distance. However, because neurons are not physically touching each other, once one neuron ends, that message should be passed across to another neuron. However, because the neurons are not physically touching, there is a gap, or what is called a synaptic cliff, in between neurons. So the electrical impulse has to be converted now to a chemical, those are called neurotransmitters, and that chemical is able to dissolve or diffuse across the synaptic cliff, and then that those neurotransmitters are detected or collected by receptor molecules, and once the receptor molecules of the target cell receives the neurotransmitters, then the message is now reconverted to an electrical impulse. And that's pretty much how these signals are sent via neurons or nerve cells. All right, and then here's a very, very common type of cell communication. We have long distance signaling. And long distance signaling is done using hormones, and those are chemicals that are produced solely for the purpose of communicating or sending messages to other cells in order for other cells to pretty much do something or respond in a specific way. So animals and plants use hormones for long distance signaling, and another name for long distance signaling is is endocrine signaling. I'm sorry, is endocrine system. signaling. And we do have an endocrine system as large multicellular organisms that pretty much help with sending these messages. So plants will release hormones that travel via the vascular tissue, which is the xylem and the phloem, or they could travel through air in order to reach the target tissues. Animals, however, we use endocrine signaling, and we do have endocrine systems depending on how complex the animal is. So, like human beings, we have a whole endocrine system in which these hormones are going to be released into our bloodstream, and then it travels from our bloodstream to the target cells. An example is, and there are several examples, but here are example. Insulin is a prime example of long distance signaling. Insulin is produced by a gland called the pancreas, and then once that insulin is produced, the insulin is sent into the bloodstream, and it will travel from the bloodstream to the target cells. Now, one thing to note with long distance signaling, and I'm going to skip over these guys. I'm going to skip over these a little bit. But one thing to know about long distance signaling is that long distance signaling requires for the target cell to have a specific receptor that can read the signal. That's very important because the message is traveling over long distances, it also means that all cells are pretty much going to be coming in contact with that signal. But however, not all cells can collect that message. All right, so let's talk about cell signaling a little bit. So, in order for cells to receive a message, interpret the message, and then pretty much respond to the message, there are steps involved in this process. And that's what we're going to go over. We're pretty much going to go over the steps of cell signaling. So there are three main stages. The first stage is reception, in which the message is received by the target cell, and the message is called ligands. So a ligand has to bind to a receptor on the cell membrane of the target cell. And then once successful reception has occurred, then we have transduction. And transduction is the process by which the signal is now converted to pretty much different chemicals. And then the last thing we're going to have is a response in which the cell will be responding in a specific way. So a cell process will be altered. So the cell is going to do something once it has successfully received and transduced that message. All right, so we're at step one. We're going to go in a little bit more detail. So reception is the detection of the message. So detection and receiving of the ligand. And remember ligand, I'm going to highlight this word right here. So ligand, where's my highlighter? Ligand pretty much is the name of the molecule or the name of the message that is sent. Those are called ligands. So those are molecules. And those molecules will be received by receptors that are found on the cell membrane or inside the cytoplasm. The cell that is receiving the message or receiving this ligand, that cell is called the target cell. So I want you guys to remember these keywords. Receptor is receptors are macromolecules that bind to a signal molecule or a ligand. So whatever it is on the cell or in the cell that is receiving the message, whatever molecule that is called the receptor. All receptors have an area that can interact with the ligand, so interact with the message coming in, and also can transmit the message to other proteins within the cell. Binding, and this part is very crucial. So I want you guys to recall how enzymes are specific to their substrate. So this is also very similar. So binding between a ligand and the receptor is a very specific process. Only specific ligands can bind to specific receptors. There are several ligands, especially over long distance communication, that are just pretty much being circulated in the bloodstream. However, they will not be received. Reception will not occur unless they have arrived at their specific receptor that is able to pretty much bind and receive that message. So once the ligand successfully binds to the receptor, we have the activation of the receptor occurring. And receptors are usually proteins, or they have protein subunits. And once the protein is now in contact with another chemical, which is our message, then that protein will undergo conformational change. Now, what is conformational change? You guys should know this because we did proteins. So the structure of the protein is changing, and therefore the function of the protein is going to change. But the protein is not going to become inactive. This protein is now going to become active. So the shape has changed in a way that the function is now a desirable function that can actually trigger a response, or pretty much the transduction process will occur before that response. Um, Allows the receptor to interact with other molecules. So once the conformational change occurs and this protein changes, and then this protein, the change in the structure of the protein changes its function, and now the proteins can interact. It's like it's switched on, and now it can interact with other molecules within that cell. And this is pretty much what initiates the transduction period. So this is the end of reception. So once the message has been successfully received by the receptor undergoes conformational changes in which the shape has changed and now the function has changed. And the change in the function now causes this receptor to now interact with other molecules. Then that is the end of reception. And now we have the transduction process occurring. Um, Before we move on to transduction, which is our stage two, please note that receptors can be embedded in the plasma membrane or cell membrane, or they can be in the intracellular part of the cell. So they can actually be in the cytoplasm of the cell. And let's discuss very briefly the difference between plasma membrane receptors and intracellular receptors. Plasma membrane receptors are proteins or molecules that have protein subunits that are pretty much embedded in the cell membrane. And these are the most common type of receptors. Um, these will bind to the ligand that are coming, so external ligands that are coming from other cells. These ligands, the reason why receptor are embedded in the plasma membrane is because the ligands that are coming in, they're polar or they're water soluble, or both. If they're water soluble, they're polar. And also they're large, or they could be charged. They could be like ions. And therefore, we know that if it's polar, water soluble or large, it needs help, assistance crossing. So that's what those receptor molecules that are embedded in the cell membranes do. Here are some examples of ligands that are large, charged, or polar. These ligands are called G protein coupled receptors. Oh, sorry, I meant to say here are examples of the plasma membrane receptors. So these are examples of the receptors, not the ligands. So the receptors that are collecting these ligands are called G protein coupled receptors, or they can also be called ligand-gated ion channels. So the ligand-gated ion channels, this is suggesting that these ones can collect molecules that are charged, so ions, molecules that are charged. And then the GPCRs are responsible for like large polar molecules. So these again are examples of membrane receptors. All right, so let's talk about intracellular receptors. So these receptors are now inside the cell, so they're not embedded in the cell membrane at all. So they're inside the cytoplasm of the cell, which means that the receptors do not have to be on the outside because the ligand or the message coming in is pretty much the opposite in its characteristics. So these messages can actually cross the cell membrane on their own, which would suggest that they are small molecules that can easily diffuse through the pores of the cell, or they are hydrophobic molecules. And examples of these, and this is what AP Bio wants you guys to know, examples of these are steroids. Steroids are hormones, guys. So remember, some hormones are steroids. So like for example, testosterone is a steroid. So if the signal molecule is described as a steroid, then you should already be able to deduce that, hey, this is going to be collected by an intracellular receptor because there is no need for help crossing the cell membrane because steroids are lipids, and therefore lipids are very small macromolecules, and also lipids are not charged, and lipids are non-polar. so they can easily cross the cell membrane. And another example is like gases such as nitric oxide. Those can also easily cross because of their size. So they can easily diffuse across the cell membrane. Um, so here's an example of a intracellular receptor. So here we have our cell membrane. Let me get back to my, so here we have our cell membrane right here. And if you look at the cell membrane right here, there are no molecules or proteins that are collecting the signal molecule. However, inside we have the receptor for the signal molecule, and that's because the signal itself can diffuse across the cell membrane. Or sometimes it diffuses across the cell membrane, and depending on what the message is and where it needs to go, it can also diffuse across that nuclear membrane to get inside the nucleus. Here's something to note. So I added this so you guys to just know and always try to recall this. AP Bio will not expect you to be able to classify any of the molecules as hydrophobic. Usually they will tell you if it's hydrophobic, or here is what they will tell you. They will tell you that it's a steroid. And if it's a steroid, this is something you need to know. So they're testing your knowledge on macromolecules, on lipids, and you should know that steroids are nonpolar, they're not charged, and they're small macromolecules. So they'll say that, hey, it's a steroid, or they'll just tell you, if it's not a steroid, they'll just tell you that it is hydrophobic. All right, so transductioners are stage two, and in transduction, the message will be converted, and then we're gonna have a signal. However, for transduction, sometimes this process does take a while, or well, not in terms of time, but several processes or several stages or several steps occur. In order for us to get a response at the end. So transduction is the conversion of an extracellular signal, which is the ligand, to an intracellular signal that will bring about a response. It requires a sequence of changes, so this is what I was mentioning, in a series of molecules known as a signal transduction pathway. So molecules are going to be changing. So there's going to be a sequence of changes that are occurring within each molecule. And that whole series of change, or that like whole cascade, is called the signal transduction pathway. And here we have the signal transduction pathway. So this process is showing us reception. Well, we have reception here. This molecule undergoes conformational change, and then it's now able to interact with this molecule. This molecule will undergo some changes to be able to interact with this molecule, and so on and so forth. So it's a series of things occurring, and that's our signal transduction pathway. All right, so some more detail about the transduction process. The signal transduction pathway, remember this is right here what we described. It's regulated by protein activities. So this whole process right here is regulated by proteins, specifically enzymes. The enzyme that is mentioned a lot, so the primary enzyme is protein kinase, and protein kinase is responsible for phosphorylation. And what phosphorylation is the process of adding a phosphate to various molecules in order for the molecules to be pretty much activated or switched on. So phosphorylation is the process of pretty much causing molecules to bind to phosphate or adding phosphate to molecules. And this is done as a result of protein kinase catalyzing that reaction. And then once this occurs, it relays a signal inside the cell. So once phosphorylation occurs in one molecule, it switches on that molecule, then the molecule interacts with another molecule, that becomes phosphorylated, and so on. There are proteins, specifically enzymes, that can pretty much remove the phosphates from molecules, and that process is called dephosphorylation. And this is performed by an enzyme called protein phosphatases. So it's more than one. So phosphatases is responsible for removing phosphates from molecules, and that's dephosphorylation. And dephosphorylation is a process that pretty much shuts down the pathway or turns the molecules off. I say this already and I'll say this again. Changes in the shape of the protein changes the function of the protein. During transduction, the signal can be amplified by what we call second messengers. And these second messengers are small non-protein molecules and ions that help to relay the message and amplify the response. And by amplifying the response, it's like you're making the response more distinct, more pronounced, or you're making the response more like it's like more abundant, pretty much. So that's what it means to amplify the response, or the response is more intense. So you're intensifying the response. Um, the amplification or the secondary molecules, we call this cyclic AMP or cyclic AMP or CAMP, and that is a very common second molecule in cell communication or the signal transduction pathway. And then here we have our stage 3, which is the response. So the response is final. So this is a final step. And in the response, this is the final molecule within the signal pathway that converts the signal to a response that will alter the cellular process. So this final molecule will now cause the cell to respond or react in a way that pretty much changes the cell's processes. So it could be, again, mitotic cell division, cell growth. Like it's causing this, it's like a call to action for the cell. So the cell is going to be doing something. An example of a response would be a protein that can alter the membrane permeability. So a protein that can cause the membrane to become more permeable or less permeable. Enzymes that will change a metabolic process, and also proteins that can turn genes on or off. So these are examples, just a few examples of responses. And these examples are not exclusive. So this is not an exclusive list of responses, guys. This is just a few of. So there are several ways in which responses there are several different types of responses, but in general, the final protein molecule can do either of these things, and then there are several other things that can also occur. Here is some quick review. I don't want, in the interest of time, I don't want to spend too much time on this because I know that you guys can always just pause the video and you can do it on your own. What are the three types of, or three stages of cell signaling? Read question again and then pause, and then that's the response. Read question, pause, and then that's the response. All right, so let's talk more about signal transduction pathways. So this is our topic 4.3. Signal transduction pathways can influence how a cell responds to its environment. They can result in changes in gene expression and function. So these are some things that signal transduction pathways can do. They can alter the phenotype or the results in cell death. So apoptosis can occur in cells as a result of these signal transduction pathway. There can be changes in signal transduction pathways. So a primary reason why changes in these signal transduction pathways could occur is as a result of mutation. So mutations in the receptor proteins. So the protein that is actually receiving the signal, and remember these are proteins made up of amino acids. Amino acids are as a result of the gene or the codon. So any changes in these amino acids can result in proteins that do not function because they don't have the right sequence of amino acids. And therefore, even though signals are sent, they just don't receive the signals, and that interferes with the entire cell communication or the transduction or the cell signal pathway. So mutations to the receptor proteins or to any components of the signal pathway will result in a change in the transduction of the signal. Some diseases are as a result of changes in these receptor molecules, and therefore they can affect how if or how that message is received. Here is an FRQ question. I want you guys to pause the video and attempt to answer this question on your own. The answer to the question is in my notes in the PowerPoint presentation, so you guys can also check that out. And then if you have any questions during class, you can always ask those questions. All right, so let's talk a little bit about receptors. So AP Bio wants you to know the two categories of receptor molecules. So these are found only in eukaryotic organisms, and there are two main types. So regardless of the type of signal or the ligand that is sent, the signal is going to be received by either or these two receptors. So we have the G protein coupled receptor, and these receptors are embedded in the cell membrane, so they're going to be extending outside the cell membrane. And then we have the ion channels, or what is called the ligand ion gated receptor, which these guys are usually going to be, they're going to be on the outside as well. So they're not specific to inside the cell membrane, to intracellular membrane. All right, so these are the two types, and this is what AP Bio wants you guys to know. So they want you to know these two types of receptors and how do they differ in terms of how they receive the signal or what type of signals they receive. All right, so let's first talk about the G protein coupled receptor. The G protein coupled receptor is the largest category of surface receptors. So most receptors are going to be G proteins. Coupled receptors are GPCRs. And these are very important in animal sensory system. The the GPCR is very similar, or there's a component of it that's very similar to ATP, and I'm going to explain why. So it binds G proteins to what is called the GTP. The GTP is an energy molecule very similar to ATP. So these G proteins are going to be binding to this GTP molecule. And here's how this works. The GPCR enzyme and the G protein are inactive until the ligand binds to the GPCR on the extracellular side. So by extracellular side, we mean on the outside of the cell. So what will happen is that we have this ligand coming in, and remember, our ligand is our signal that was sent from the other cell that wishes to communicate with the target cell. So this ligand is coming in, and this ligand is now going to activate this GPCR. And what the ligand does is the ligand has to first be a perfect fit. So remember, it's specific to its receptor. This is inactive. So this is the inactive form. So ligand has not successfully binded yet. This is the GPCR in its inactive form. Here we have GDP, and the DP stands for diphosphate. And then we're going to have an inactive enzyme. So this enzyme is also inactive. And then let's switch a slide to see what happens when we have successful reception occurring, how these molecules change. All right, so once we have a successful reception process occurring in which we have our ligand now binding to or GPCR, this causes the cytoplasmic sites to change shape. So once we have this successful binding, we have activation of our GPCR, and then we are going to have this active G protein binding to this GTP, and well, not binding to the GCP, but it's going to convert that GDP to GTP. So we're going to have that conversion occurring. Now, once we have that successful reception occurring, and then we have all these changes occurring, the product of this molecule, this G protein that is also now active with our GTP, that is now used to activate the enzyme. And then once this enzyme is activated, it pretty much amplifies the signal. And then once it amplifies the signal, then we have the signal pathway occurring, and then we're going to get a response. So there are a few things that will happen with with G proteins in order to get a response. And it functions differently than ion channels. And we're going to get into to ion channels. All right, so ion channels are ligand-gated channels. So these ion channels, as the name suggests, it is responsible for collecting ligands that are ions themselves, or ligands that are responsible for controlling the concentration gradient of ions. And this type of receptor plays a huge role in the nervous system, in which nervous impulses or signals are converted to neurotransmitters, which are ions, and then sent back as electrical impulses. So ions do play a huge role in the nervous system. So the receptors act as a gate for ions. When the ligand binds to the receptor, the gate either opens or closes, allowing for the diffusion of specific ions. Um, and this initiates a series of events that leads to a cellular response. And I have a diagram on the next slide that I'm going to show you. All right, here we go. So this is ion. This is like a ligand ion gated receptor. So here we have our neurotransmitter, which is our ligand right here, and it is required to open the ion channel. And the ion channel is this passage right here. So right now the ion channel is closed off, in which we do have a higher concentration of calcium ions on the outside of the cell, and a higher concentration of potassium ions on the inside of the cell. So there is a concentration difference. And then we also have our neurotransmitter, which is our sodium ions. So the ion channel is closed. Here we have our ligand. Our ligand will successfully bind to the receptor at the receptor site. And then once it successfully binds to the receptor at the receptor site, what you're going to have is, so this is a successful binding, what you're going to have is that the gate opens, and then we're going to have sodium ions um, going in. So here we have our sodium ions going in, and then we're going to have our potassium ions coming out. And then also we're going to have some calcium ions also going in. So it pretty much opens the gates. And then also if this ligand is removed, then the gate closes. So it pretty much mediates the concentration of ions in the cell. And this, again, the key thing is this serves a role in the nervous system. And that's all for this lecture, guys. So I covered this in less than 40 minutes, but remember, this was 3 topics in one. So it means that we have 2 more topics to go.